In today's video, we're going to be taking a look at striated muscle and the neuromuscular junction for A-level biology. Now, the first thing that we need to cover are the two different filaments that we have in striated muscle. So we have actin filaments. Now, an actin filament is composed of actin, which is a globular protein that polymerizes with other actin molecules to form a double helical structure. As we see here, it's double helical. We then also have tropomyosin molecules. Now, a tropomyosin molecule is composed of two long polypeptide chains that coil around each other, as we see with this blue pen. And then we also have troponin molecules. Troponin is a globular protein, and the troponin molecules attach to the tropomyosin molecules to form a troponin-tropomyosin complex. Now, together with the troponin, tropomyosin, and actin, we have an actin filament. Now, the tropomyosin molecules will actually block the binding site of the actin molecule so that a ectomyosin crossbridge cannot be formed. So this means that the myosin heads will not be able to attach to the binding sites on the actin filament. Now, the actin filament is thinner than the myosin filament. So the myosin filament is thicker than the actin filament, and it is composed of long rod-shaped fibers that have bulbous heads. As we see here, it's a rod-shaped fiber and it has this protruding bulbous head. The tails, as we see here in the full myosin filament, the tails wrap around each other and they form the filament. So the filament, the myosin filament, will have all of the tails wrapped around each other, and these bulbous heads, also known as myosin heads, protruding outwards from the myosin filament. Now over here, I've drawn a whole muscle, and within the whole muscle, we can see that we've got these bundles of muscle cells, within which are the single muscle cells. Now we can see here that we have these little points, and these are the blood capillaries. Now each muscle cell is composed of hundreds to thousands of myofibrils, and myofibrils have darker and lighter regions, and this pattern is the reason why the muscle is actually called striated muscle, okay, because it has this pattern of dark and light. Now, the reason why we have these darker and lighter colored regions is because of the anisotropic bands and the isotropic bands. Now, the anisotropic bands are those that are dark, right? And if we take a look, so dark, the second letter is A, that means it's anisotropic. And this is caused by overlap of the actin and myosin filaments. On the other hand, the isotropic bands are the lighter regions, and we can see here in the word light, the second letter is I, so it's isotropic. Now, if we take a look here at a sarcomere, so a sarcomere is one single unit of a myofibril. Now, we can see here that we have the myosin filament, which is this thicker filament, and we have the actin filament, which is this lighter filament. Now, we have these Z lines. So Z lines are in the middle of isotropic bands. So in the middle of these lighter colored regions are the Z lines, and the Z lines anchor the actin filaments. Okay, so in the isotropic bands, only the actin filament is found. There is no myosin. So we have the Z line, which is a protein that is anchoring the actin filaments. In these darker regions, in the anisotropic bands, within them, in the middle, is the H zone. So this is the lighter colored zone within an anisotropic band. And in the middle of that H zone is going to be the M line. And this is going to be a protein that anchors the myosin filaments. Now, let's quickly take a look here. So this is, as we said, a sarcomere. Now, the region where there is overlap will cause a dark color. Okay, so we can see here, this is an anisotropic band, and we have a darker region, which is caused by this overlap. We then have a lighter color region, which is because there is no overlap of any filaments. And then we have another darker colored region because the myosin and actin filaments, again, overlap. We then have only actin, and because there is no overlap there, it is simply just going to be light colored. Now, between each of the Z lines, so between adjacent Z lines, is one sarcomere. Now, I really want to make it understood that the anisotropic bands are dark because of overlap. Okay, we see that there is overlap between the filaments here, and so it is a dark color. Here, there is no overlap. Despite it being myosin only in this region, it is light colored. Okay, now I want to make sure, because in isotropic bands, we only have actin. So you may think that, oh, in anisotropic bands, the dark color is because of the myosin, but that's not the case. As we see here in the H zone, there is still this lighter colored region, even though there is only myosin, and that's because there is no overlapping. Now let's take a look at the neuromuscular junction. So there'll be an image now on the screen. The neuromuscular junction is basically the point of communication between a presynaptic neuron and the muscle. Now, there are going to be involved neurotransmitters, like, for example, acetylcholine. There will be involved calcium ions. So calcium ions so an action potential will come down the presynaptic neuron, which will depolarize the presynaptic membrane. This is going to cause the opening of voltage-gated calcium ion channels, causing calcium ions to enter into the synaptic knob, right into the presynaptic neuron. Now, within the synaptic knob of the neuromuscular junction, there are these synaptic vesicles that contain neurotransmitters like acetylcholine in this situation. With the uptake of calcium ions into the synaptic knob, the synaptic vesicles will then fuse with the presynaptic membrane, releasing the neurotransmitters into the synaptic cleft. The acetylcholine that is now in the synaptic cleft will attach to the ligand-gated sodium channels in the sarcolemma. That will cause the opening of the sodium ion channel. So sodium ions will enter the sarcolemma and they will depolarize it. Now, if enough sodium ions enter, if there was enough neurotransmitter, enough acetylcholine, an action potential will be stimulated. Now, what's then going to happen, we're now moving on to muscle contraction. So if an action potential is stimulated, then what happens is the sarcolemma, so the membrane of the muscle cell, has invaginations which fold deep into the muscle fiber. And these invaginations are called transverse system tubules. So if there is an action potential in the sarcolemma, it will travel deep into the muscle fiber via the T-tubules, where it will eventually reach the sarcoplasmic reticulum. Now, as I discuss in the muscle contraction video, the sarcoplasmic reticulum up until now has been actively uptaking calcium ions. But because of this action potential, it opens the calcium channels. Okay, so then the calcium ions will leave the sarcoplasmic reticulum and go to the sarcomere. Now, what they will then do there is that they will attach to the troponin on the troponin-tropomyosin complex, which will cause the displacement of the tropomyosin molecule. This means that the tropomyosin molecule will no longer be blocking the binding site on the actin, and that means that the bulbous heads of the myosin filament can attach to the actin binding site, forming an actomyosin cross bridge. Then, with the power stroke, the actin filament will be able to be moved along the myosin filament. And so what happens in muscle contraction is that the Z lines will come together. So they will narrow. This means that the isotropic bands will become narrower, and this also means that the space where there was just myosin will now also have actin overlapping the myosin filaments, and that was the H zone. So that also means that the H zone will become narrower during muscle contraction. Now, muscle relaxation will occur when the sarcoplasmic reticulum will actively uptake the calcium ions back into it, and this means that the tropomyosin molecule will then recover the actin binding site, and so a ectomyosin crossbridge will not be able to be formed, and so muscle contraction will not take place. Now, this is also known as the sliding filament model, because as a result of the power stroke that the myosin heads do to the actin filament, the actin filaments actually slide along the myosin filaments. And in the sliding filament model video that I did, I actually go into more depth on how this actually works. So that's really it. I really just want to reiterate. So we, in the A band, so in the anisotropic bands, in the middle where there is that lighter region, it is called the H zone. So this is where there is just going to be myosin filaments. There is no actin to overlap it, which creates the dark color. And in the middle of the H zone is the M line. And this is composed of a protein that anchors the myosin filaments. In the isotropic bands, where it is light colored and there is only actin filament found, we have the Z line. And this is a protein which will anchor the actin filament. And again, I want to reiterate that the dark color is caused by overlap, not because there is myosin. Okay, so it is only because there is overlap of the actin and myosin filaments. So that's it for this video. I really hope you enjoyed it. If you did, leave a thumbs up. Take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thanks very much for watching, and I hope you have a great day. Bye-bye.